Okay, Assalamualaikum and good morning, class. Can you hear my voice?
Good morning, Dr. Yes,
Class.
yes, doctor, we can hear you.
Assalamualaikum and good morning. Can you hear my voice?
Yes, doctor. Yes.
Oh, sorry, my. Okay, thank you for your response. Okay. So before I start, as usual, as a reminder, I have set an attendance in the Putra Blast. I hope that if you cannot enter, you or any one of you that cannot enter the live sessions is okay, because I have also recorded the lecture. It's just that it's very important for you to say in the attendance. I got an information from the course coordinator, BPM 3340, from Prof. Suri. She stated that based on previous lecture performed by Prof. Dato Azmi, some of you not attend the class, like if I'm not mistaken, 3 times. So she reminded me to also remind all of you, the attendance is very crucial, because the ESMP system will record your attendance. So basically, if your attendance is less than 80%, by default, the system automatically will prohibit you from enter the final exam. So this is very crucial. So for my part, it's okay if you don't attend the live sessions. The most crucial part is you need to sign in your attendance, okay? Because I understand some of you might have connection problems. So that's why I record my lecture. Okay, so for today, as usual, when I give lectures, you are free to interrupt if you have any questions or you need further clarification from me. So for today, the topics that I would like to cover is arbovirus. Arbovirus is actually an acronym for R is for arthropod, B for born, and virus for virus. So basically, this arthropod-borne virus or arbovirus is actually transmitted via vectors, and these vectors are varies. They can be mosquitoes, ticks, or sandflies. And what are the role of these mosquitoes is actually is termed as vectors. So what is vectors mean? So vectors, basically, they carry the pathogens and transmit the pathogens from one host to the other host. And most of the time, the vectors won't get infected, but in some circumstances, some of them might be affected, causing low lifespans of the vectors. So basically, the examples of vectors can be mosquitoes, and in arbovirus, examples of diseases caused by mosquito-borne infection is Japanese encephalitis, dengue, yellow fever, St. Louis Louis encephalitis, West Nile fever, chikungunya, Eastern equine encephalitis, Western equine encephalitis, and Venezuelan equine encephalitis. And actually, there are a lot. You will see later on in my slides. And the examples of disease that is transmitted by ticks is also known as Cameroon Congo hemorrhagic fever. And in terms of flies, for example, sand flies, they also able able to carry Rift Valley virus, which causing Rift Valley fever. So this is the examples of mosquitoes, Culex mosquitoes, Aedes mosquitoes, and this is the examples of assorted ticks, and this is the example, the picture of sandfly. Okay, so basically, I want to move my picture here. Okay, alright. So basically, in general, arbovirus mainly is RNA virus. Okay. A lot of arbovirus or viruses classified under arbovirus are RNA virus, and the only member of DNA virus is known as SV virus. Okay, and for RNA virus, as you know, you have single-stranded positive strands, single-stranded negative strands, and double-stranded RNA. In the case of Rio virus, um, orthomyxovirus, and rhabdo, as well as SV virus, you will have a specific lecture later on, which will cover these topics. Okay, for the purpose of today's lectures, I will cover the arbovirus caused by Toga viridae family, Flaviviridae family, and Bunya viridae family. And you can see in each family, there are specific genus that causing this arthropod-borne virus. Okay. Okay. This is the general classification of arbovirus, the one that I would like to cover for today. So under Toga viridae family, you have two genus known as alpha virus and ruby virus. And you will see later on, you will appreciate that some of these family genus under this family, they are non-arthropod borne. Okay, so basically for today's lecture, because I covered Toga, Punya, and Flaviviridae family, so because of that, I will also cover the non-arthropod borne virus. Okay, so for Toga viridae, you have alpha virus. So examples of viruses under alpha virus genus are Eastern equine encephalitis, Western equine encephalitis, Venezuelan equine encephalitis, Getah virus, chikungunya virus. And another genus is Ruby virus. Uh, this is actually non-arthropod borne, so I need to amend it later on. And it is actually the virus in human which causing rubella infection. Next is the Bunya viridae family, where you have the arthropod-borne and the non-arthropod-borne. Under arthropod-borne virus, you have three typical genus of Bunya viridae known as flavivirus, nairovirus, and Bunya virus. Okay, this is the examples of virus. Rift Valley fever caused by Lebo genus. Cameroon Congo hemorrhagic fever caused by nairovirus. And Acabane virus and Cache Valley virus is caused by Bunya virus. On top of that, under Bunya-Viridae family, you have non-arthropod-borne, which is caused by hantavirus genus. And the examples of viruses under this hantavirus genus are Hantan virus, Humala virus, and Sol virus. Okay. Meanwhile, other families are under arthropod-borne virus is known as Flaviviridae. You also have two main classification, which are the arthropod-borne and the non-arthropod-borne. Under arthropod-borne, you have two genus, namely Flavivirus and Pestivirus. And this flavivirus, you can further subdivide it into mosquito-borne and tick-borne. And for mosquito-borne, most of you, I think you are aware, the examples of flavivirus mosquito-borne are JE, Japanese encephalitis virus, West Nile virus, dengue, yellow fever, and you have also St. Louis encephalitis virus and Murray Valley encephalitis virus. And under tick-borne flavivirus, you have an example of looping eel virus. Okay, and another genus under arthropod-borne is known as pestivirus, which cause infection mostly in animals. The name of the disease is actually bovine viral diarrhea. I need to correct here. I should write here the name of the virus, not the name of the disease. So here it should be bovine viral diarrhea virus, BVDV. Okay. Okay, and then you have border disease and classical swine fever or CSF. And this Flaviviridae family also you have there is also non-arthropod-borne virus which is classified under genus Hepativirus, causing hepatitis C and G particularly in human. So basically, what is happened when this for this slide, I'm giving the examples of mosquitoes. What happened actually when the mosquitoes enter the host, it will inoculate the virus via their saliva into the host, and these most of the time I am giving you the general pathogenesis because for each virus they have their own specific pathogenesis. So basically, what happens is that when the virus is entering the host body. So the first cells that will uptake the virus is known as Langerhans cells, which is a type of dendritic cells. And what happens is that the virus will go to lymph nodes and will travel through the lymphatic system and bloodstreams, causing viremia, and it will go to the tropisms. Okay, and tropisms of arthropod-borne, mosquito-borne, tick-borne, or flies-borne are actually varies. It's very specific for each virus. So I'm giving here an examples of some of the viruses. They have neurotropic tropism. Some of them congenital tropism. Some of them can cause a visceral disease. Okay. So generally speaking, most of the infections of arthropod-borne virus, this is an typical examples under flavivirus infection. 70 to 80% of the infections are asymptomatic, whereas approximately 20 to 30% is associated with viral disease or symptoms such as febrile illness, headache, also have vomiting, muscle pain, and less than 1% is associated with neurological disorders, where most of these viruses, most of them will have neurotropic tropism. And the manifestations of these symptoms most of the times is highly dependent on the immune system of the host. Okay. So there are few factors that favors the maintenance of arbovirus in the environment. The first important factors is the host. The host can be amplifier host, it can be dead end host, it can be the reservoir host. So most of the time, how does this arbovirus maintain their presence in the world is basically they must achieve high viremia, which is sufficient for it to transfer to the other host during the blood meal. So basically, under these host factors, I would like also to share the general transmission cycles of vector-borne virus. As I mentioned earlier, you have reservoir host, you have amplifying host, and you have dead end host. So basically, how this virus maintain their presence is that by you can see based on the illustration of these cycles. There are various cycles where this mosquito bonds is actually transmit the virus. Mosquito bonds disease is being transmitted via they are known as enzootic cycle, epizootic cycle, or epidemic cycles. Under the enzootic cycles or sylvatic cycles, also known as jungle cycle, this is the natural transmission of mosquito-borne virus, which is actually is happen in the jungle where the transmission is occurring between the mosquitoes and the wild animals, which is leading to the amplification of the virus. And most of the time, why we term is as enzootic, it means that it is prevalent in the animals, in the wild animals, which they exist in nature. Okay, whereas there is also cycles known as epizootic cycles or rural cycles, which mainly involve domestic animals. So, what these domestic animals their role is that they are the amplifying host. So one of the typical example of epizootic cycle is in the case of Japanese encephalitis, where swine, where we call it rural cycle because most of the time these domestic animals are being bred in the rural area. So this swine is their role is as amplifying. They amplify the virus, meaning that they have sufficient viremia when the mosquito bite them, and when this mosquito transfer the virus to other animals, it's capable of causing the virus to undergo replications in the new host. Okay, and on top of that, you have also epidemic cycle. Epidemic cycles meaning that the occurrence of the disease is quite severe, quite tremendous because it involves more geographical locations and it involves more animals. Okay, this is known as epidemic cycles. Most of the time, these epidemic cycles is known as urban cycles, where human is the responsible host. They are the source of infection. They are the amplifier of the disease causing high level of viremia. The typical examples, for example, in Malaysia is dengue virus. And most of the time, these three cycles, you have a dead end host or incidental host, where in the dead end host, why they are known as dead end host is because of that they don't have enough viremia, insufficient viremia to cause further replication of the virus to other hosts. So under this dead end host, the examples is snail virus, Eastern equine encephalitis, and Western equine encephalitis. So this is basically how the host play significant roles in maintaining the vector-borne virus in the environment. On top of that, you have the vector itself, the presence of vectors. Normally, in the case of mosquitoes, a single mosquito, they have ability to carry more than one virus. In the case of Aedes, they can carry dengue, yellow fever, chikungunya, and Zika. And you have also Culex mosquitoes where they can carry West Nile virus and Japanese encephalitis. virus. So, and these mosquitoes, they breed very easy. A lot of viruses where their bodies exposing them very abundant in the environment, and they have multiple species, Aedes, and Meninges, Culex, and so on. So basically, what is the role of mosquitoes swarming? Uh, how does this mosquitoes is actually transmitting vector-borne virus? I would like to share with you a YouTube video. Can you hear, class?
Yes,
Yeah,
doctor.
Yes, doctor.
Okay. Feel free to ask questions if you don't understand or you need any or you need clarification. And that's it.
Hello everyone, I'm Vincent Racaniello and this is Virus Watch, the weekly video report on what is happening in the amazing world of viruses. Can you name 5 viruses that are spread by mosquitoes? Zika virus should be the first virus you think of, followed by dengue virus, chikungunya virus, yellow fever virus, and West Nile virus. These viruses have all been in the news a lot, so you should recognize them. Let's take a look at how mosquitoes transmit the virus from one person to another person. The name mosquito, by the way, is Spanish for little fly. Mosquitoes are winged insects with 6 legs and tube-like mouth parts called a proboscis, which is used for feeding. There are over 3,500 different known mosquito species, and it's estimated that mosquitoes have been on Earth for nearly 200 million years. Both male and female mosquitoes feed on plant juices, but only female mosquitoes take blood meals. And that's because female mosquitoes need blood to produce their eggs. The female mosquito detects a potential biting victim by first following trails of carbon dioxide, which of course we exhale with every breath. Then the mosquito homes in on a variety of organic compounds that make up what we call the human odor loop, such as lactic acid in the skin and ammonia in human sweat. By the way, the mosquito DEET works because it blocks the odor receptors that mosquitoes use to sense these chemicals. After the mosquito lands on the skin, she probes with her proboscis to find a suitable spot, then pierces the skin. The mosquito mouth is made up of 6 different parts, but only 2 of these actually penetrate the skin. Here's a diagram of human skin, which is made up of an outer layer called the epidermis, and underneath it, the dermis. The mosquito inserts her proboscis into the dermis because that's where blood vessels are found. The mosquito then searches with her proboscis for a blood vessel. This process is shown in this movie of a mosquito on the skin of a mouse. You can see the proboscis, the long, flexible brown tube, as it's moved back and forth by the mosquito on the surface looking for a blood vessel. During this probing period, the mosquito is injecting saliva into the host. Saliva contains a collection of chemicals which include anticoagulants. These prevent blood clotting, vasodilators to keep the blood vessels wide, and anesthetics to prevent us from sensing the mosquito as it slides the proboscis around in our skin. We would slap the mosquito and kill it otherwise. You can see in this movie that the mosquito finally finds a blood vessel. The proboscis slips in to the vessel and begins to draw blood. It's amazing that the mosquito applies so much suction that the blood vessel is actually becoming thinner. As the mosquito draws in blood, it fills her midgut, and that's what you might call her stomach. The blood cells are kept, and the plasma, which is the liquid portion of the blood, is discarded via the anus. If there is virus in the blood of the host, a condition that we call viremia, then of course the mosquito would be ingesting the virus along with the blood. Over the next few days, the blood cell proteins are digested, and then the female lays a batch of eggs. During this time, the virus replicates in the mosquito midgut and enters the circulatory system, where it can then spread throughout the mosquito, reaching the salivary glands. For West Nile virus, it takes 7 to 10 days for virus to appear in mosquito saliva after the mosquito has taken a blood meal. When the female mosquito is ready to make a new batch of eggs, it seeks another blood meal. This time, when the mosquito is probing and depositing saliva in the skin, along with the saliva comes virus from the salivary gland. And that is how mosquitoes transmit viruses from one host to another. You can see from this series of events that there are two main requirements for transmission of a virus by a mosquito. First, the virus must cause a viremia in the host so it can be taken up by the mosquito while the mosquito is ingesting blood. Second, the virus must replicate in the mosquito and make its way to the salivary glands from the midgut. These two requirements limit the number of viruses that can be transmitted by mosquitoes. For example, influenza virus is not transmitted by mosquitoes, in part because it doesn't cause a viremia. HIV does cause a viremia, but that virus cannot replicate in the mosquito. Sometimes mosquitoes transmit viruses mechanically without the need for virus replication in the mosquito. An example is myxoma virus, which infects rabbits and is spread by mosquitoes. While biting an infected lesion on the rabbit's skin, the mosquito proboscis is contaminated with virus, which is then transmitted to the next host. Amazingly, components of mosquito saliva also seem to stimulate virus replication in the host. This observation has been made for dengue virus and West Nile virus. Mosquito spit may also stimulate Zika virus replication. In case you're wondering, the mosquito has no particular desire to transmit viruses. Viruses are parasites that are just effectively transmitted by this route. In fact, in many cases, virus infection of mosquitoes may be bad for the mosquito. It can shorten their lifespan or their ability to produce eggs. That's Virus Watch for June 8th, 2016. For more in-depth discussions about viruses, check out our science show. This
basically what happened when mosquitoes enter was and how the virus is being transmitted by the mosquitoes. Okay, so this is one of the factors that favors the maintenance of the virus in the environment. Other than that, in terms of geographical distributions, mostly temperate regions are the virus actually present. It circulates most of the year, which is often with a broad seasonal peak. It depends on which kind of tropical areas, and in the temperate countries, the virus is transmitted between the vector and the vertebrate host is mostly during the warmer months. Okay, and most of the time during the winter season, seasons or all the months, the peak of arbovirus infection is becoming low in the temperate regions. However, how does this mosquito survive all the months? Interestingly, there is known as overwintering of the eggs. So these eggs will delay their hatchings over the winter periods, and they will hatch during the warmer seasons. This is quite interesting how the mosquitoes survive in the temperate climates. So you can see from the geographical distribution here, most of the distributions of mosquito-borne virus, for example, in the case of Zika, you can see this is very prominent in the temperate region, in the tropical regions, which including Malaysia, and in the case of dengue virus, some of them in the temperate regions, and West Nile virus, it is all over the world because it was believed to originated from West. Okay. So apart from that, human behavior also have increased the survivability of this virus in the environment. Basically, human plays significant roles in the emergence and re-emergence of arboviral disease. Nowadays, if we talk about before COVID-19 pandemic, in the modern travel and trade, it is very quite common for people to travel around the world. And interestingly speaking, during travel, these mosquitoes also might also travel along with us. If you, some of you or most of you might have experienced when you enter plane. When, for example, the plane for example, from Japan or from US or UK landed in Malaysia, you will see when you enter the plane, you will see the stewardess, they will spray like insecticides. So basically, because this mosquitoes is very small, they can enter the planes when the planes landed in Malaysia. So but somehow there must be some of the mosquitoes they survive. Okay, this is a typical examples of how our behavior is actually favoring the mosquitoes. And some of the mosquitoes, they have anthropophilic behavior, whereas they love humans blood. So because of that, in the case of human effect of this infection in humans is quite tremendous and deadly because these mosquitoes simply just love human blood. Okay, and some of the mosquitoes also tend to like suck bloods of the animals. Okay, because most of the time the mammalian blood is quite warm. Warm situation will favor the breeding of the mosquitoes. Apart from that, environmental factors such as agricultural development, urban expansion, which subsequently lead to the overpopulation, and then most of the time this urbanization is unplanned, accompanied by crowded condition, and will lead to the inadequate water supply, also shortage, which is quite familiar, right? Which is happening in our country right now. And there is also waste management problem. And these factors, these circumstances collectively provide ideal breeding sites for the mosquitoes. Apart from that, climate changes or global warming also has played significant roles in the emergence and re-emergence of the mosquito-borne virus or arthropod-borne virus. Okay, so now I would like to proceed to the each family of the virus. So I will start with Bunya virus. So basically, Bunya virus, the virions are in this spherical shape. Okay, most of the arthropod virus, they are envelope virus. And this Bunya virus, the diameter is 80 to 100 nanometer in diameter. So this virions of Bunya virus, they have glycoproteins, spikes, okay, but they don't have matrix proteins like a flavivirus. Okay, their nucleocapsid has three segments, which are their nucleic acid has three segments, which are the L segments, M segments, and S segments, which is actually S is actually means small, M medium, and L for large. And the total size of the genome of Bunya virus is approximately 11 to 19 kb. Okay, and because they are RNA virus, so their replications occur in the cytoplasm, and during the replication process, the budding of the virus occur in the Golgi vesicles. And most of the time, the infections of Borneo virus is cytocidal in the vertebrate cells, but non-cytocidal in the invertebrate cells, which is in the mosquitoes or tick. So basically meaning that they were causing effects in the mammalian host body to the human or to the animals, but no effects after replication in the arthropod. Okay, so this is the examples of major pathogens under Punjaviridae family. So as I mentioned earlier, under Punjaviridae family, you have flavivirus genus, and the common virus that cause flavivirus infection is known as Rift Valley fever virus. And in terms of geographic distribution, this virus mainly found in Africa, and the arthropod responsible for the infection is mosquitoes, and the target host is or the amplifier host is the sheep, cattle, buffaloes, and human. Which means that when we term that them as amplifier, meaning that they capable to amplify the virus and causing viremia. So basically, Rift Valley fever causing disease, systemic disease, hepatitis, and abortion in cattle, sheep. Okay, and in human, they cause flu-like illness, hepatitis, and hemorrhagic fever, and retinitis. So far in Malaysia, there is no any report of Rift Valley fever virus in our domestic animals, but we are not sure about the serological evidence because most of our animals are import animals, so there is a need for studies to carry on to know the status of this virus infection. Under Nairovirus genus, we have Nairobi sheep disease virus, which is also not familiar in Malaysia. It oftenly occur in Eastern Africa, and also the Crimean Congo hemorrhagic fever also is prevalent in Africa, Europe, and Asia as well. I have read a paper where in Malaysia there is Crimean Congo hemorrhagic fever, but there is serological evidence. So I'm not sure. Probably we might have the clinical case, but because the data is very limited, so we don't really know. Okay, and mostly the animals affected are sheep, cattle, and goats. And if the infections involve the animals, most of the time it is mild, whereas in the humans it is quite terrific, where it causes hemorrhagic fever and hepatitis. Okay, another genus under this family is Bunya virus, which is example is Acabin virus, which is also not really prevalent in Malaysia. They are prevalent in Australia, Japan, Israel, and Africa. And the arthropod responsible for the transmissions are the mosquitoes and culicoides. And the targets or amplifier hosts are cattle and sheep. And diseases in animals is characterized by arthrogryposis and and hydranencephaly. So in human, it didn't affect human. So this is typical manifestation of arthrogryposis where the the calf were born with congenital anomalies, and we can see that the infections mainly involve nervous system. And hydranencephaly means that there is absence of the brain hemisphere. So the most of the time the animals will show nervous signs. Okay. This also cache valley virus also causing the same manifestations, basically very prominent, or disease usually occur in United States. Okay, you have also Hunter virus, which is under the genus hantavirus. The example is Hantan virus, which is prevalent in China, Russia, Korea, and this hantavirus is non-arthropod-borne. So, like Hantan virus, the amplifier host or reservoir is the mouse, and most of the time the mouse is will not be infected, but the disease in human is quite severe, where it causes hemorrhagic fever with renal syndrome. You have also Pomala virus, Sol virus, which are prominent, which is prevalent in worldwide. Also in the Rattus species, which is in the rat, and it also lead to hemorrhagic fever with renal syndrome. Remember in class, although some of the virus you have not hear happening in Malaysia yet, but we don't know. As I mentioned earlier, some of them might have antibodies to the virus. And remember, most of these viruses are RNA virus, and because they are RNA virus, they are very prone to mutations. So we don't know. Because this highly mutated virus, they easily being transmitted. Probably some of these viruses are sporadic. Probably it happened in the very isolated place. But in the as you can see in the case of COVID-19, which is at first it's very sporadic. It's happening in the Wuhan city of China, and you can see how the disease very fastly, rapidly being transmitted the whole world. Okay, but I just want you to bear in mind that this virus might be present, or the outbreaks also might also happen in Malaysia, but the severity will not be as severe as coronavirus. As I mentioned to you in the first lecture, family of virus under Coronaviridae and Otomyxoviridae, they are typical examples of virus that will undergo rapid mutations, and the effect is quite tremendous. Okay, so next we move on to the Toga virus. The variance also is spherical. Normally it has very uniform appearance. Okay, it's also an envelope virus. So you can see here the unique appearance of the virus, where you will have this spike protein and the capsid protein, and this spike protein will make the outer structure of this what you call it is the glycoprotein, and at the inner part of the virus, this is the capsid protein. Okay, so they are basically 70 nanometer, and the capsid is nucleocapsid is 40 nanometer in diameter. Okay, it's also RNA virus. Their genome size is basically, this is their genome, basically 9 to 7 to 11.8 kilobyte in size. Okay, and they have the structural proteins and the non-structural proteins. Okay, right, and as you know, because it is RNA virus, the replications occur in the cytoplasm and their maturations occur via budding from the plasma membrane of the host cells. Okay, so these are the examples of viruses under toga viridae. You have Eastern equine encephalitis, Western equine encephalitis, and Venezuelan equine encephalitis, which is prevalent in America or US. So basically, the animals affected are horse, okay, and normally humans are the dead and host. So basically, the horse will manifest encephalitis manifestation or clinical signs, and they are mainly mosquito borne. And interestingly, there is also Getah virus under this family. It also affects horse, manifested by febrile illness, and it is prevalent in Southeast Asia. So if you want to know more about why the virus is known as Getah, you might try want to search through. Okay, so you have also alpha virus, uh, which is known as chikungunya virus under this toga virus family. Okay, so basically it causes encephalitis, fever, malaise, and arthritis. So as you know, chikungunya is quite prevalent also in Malaysia. If I'm not mistaken, last year there are several outbreaks in Perak in humans. So mainly it is involving like most of the infections involving are uh, the joints. So the characteristic of chikungunya fever is like have bent joints, especially at the legs. Okay, so and then you have flavivirus. Okay, flavivirus also spherical in shape. The virus is spherical, and the diameter is approximately 40 to 60 nanometer, and it has lipid layer. Most of them are envelope virus. So as you know, envelope virus, they are made up of lipid. So it has indistinct spikes or speculomeres. Okay, this is their peplonians or their spike protein, glycoproteins, and they are surrounded by spherical nucleocapsid. Okay, in terms of their genomes are contain RNA, linear, positive strands, and single stranded. And most of the time, flavivirus, their genome size is approximately 11 kb. Okay, for pestis, this is for the non-arthropod-borne like pestis and hepativirus, the genome size is quite varies, which is for pestis it is 12.3 kb, and for hepativirus is 9.6 kb. So as usual, because it is RNA virus, so the replication took place in the cytoplasm, and the maturations occur in the intracytoplasmic membranes without evidence of budding. Okay, this is the examples of viruses under genus Flavivirus. You have genus Flavivirus, Pestivirus, and Hepativirus. So you have Japanese encephalitis. Okay, the reservoir host is swine and wild birds. Okay, so okay, the reservoir host is swine and wild birds, and the arthropod that involved is Culex mosquitoes, and normally the infections is causing abortion or neonatal disease in animals or encephalitis in humans. Japanese encephalitis is also quite prevalent in Malaysia, especially in Sarawak. I think you also familiar with the case of one of the juvenile, or if I'm not mistaken, one of the students from Unimas affected by JEV, and if I'm not mistaken, it led to death. So you can check. So actually, JE is quite prevalent in Malaysia, endemic in Malaysia, like dengue. Okay, so the geographical distribution of JE is mainly Asia. Okay, you have also Murray Valley encephalitis virus, which is prominent or prevalent in Australia and New Guinea, and it is characterized by encephalitis in humans, and the reservoir host is also wild birds, and the mosquitoes involved also from the Culex species. We have also St. Louis encephalitis virus, which is common in US, Canada, and South America. So basically, this involve the temperate regions, and the reservoir host is the wild birds. Okay, you can see that wild birds is actually they are the reservoir for lots of virus. Okay, in the case of avian influenza, they also one of the reservoir hosts in the case of Newcastle disease, also the reservoir host in the case of West Nile virus infection, JE. Okay, so wild birds play very significant roles in the maintenance of virus in nature. Not only wild birds, wildlife especially. Um, you have macaques, you have bats. Okay? This is typically most, as I said previously, earlier, most of the natural transmission occur in the wildlife. So does in the case of flaviviral infections. Okay, so you have also in Africa Westerborn virus affecting sheep. So it causes generalized infection and confusion. You have dengue virus. There are 4 genotypes of dengue. The very severe one is the genotype that causing hemorrhagic fever, which lead to death. Okay, so the reservoir of dengue was initially from the macaque. Okay, from the non-primate host. Okay, so basically dengue, the hosts are the vectors are mosquitoes, especially Aedes species, and the infections are not really in animals. It's mostly affect humans. None of the no reports yet have demonstrated dengue virus in animals. So basically in human, you know, right? Because it's very, very prevalent in Asia. It is characterized by fever, rash, myalgia, hemorrhagic fever, and it is occur at the tropical countries worldwide and in the subtropical countries uh, um, occasionally. Okay, and then you have West Nile virus, which is also transmitted by wild birds, and incidental hosts are the horses and humans. And some of the time, birds also, if they are immunosuppressed, they also easily also become infected with West Nile virus. And the vectors involved in the transmission of West Nile virus are mosquitoes, mainly Culex species, and it is characterized by fever and encephalomyelitis. So the distributions of personal virus is in Africa, Middle East, North America, Central America, and South America. As for Malaysia, because my research area is on personal virus, we have seen evidence of we have seen serological evidence of Western virus in pigs, in wild birds, in horse, in cattle, in small animals, and uh, Interestingly, we have also demonstrated presence of West Nile virus RNA in horse, in wild birds, especially the migratory birds and the resident birds, because these migratory birds if you see if the reservoir hosts are migratory birds or wild birds, most of the time they are wild birds. Most of the time they are migratory birds where they just migrated around the world. They have their own. Most of these birds they have people fly away from the hemisphere, northern hemisphere to the southern hemisphere. So basically what happens that when they migrate they have several landing areas, and in Malaysia there are few landing areas of these migratory birds. And what we did was we went to this migratory birds landing area and we sampled the wild birds, and turned out to be that the migratory birds have this West virus. And how we want to know whether the residents wild birds that already have their place in Malaysia, where they most of them don't migrated, they just live in the residential area. So what happens that we also take the samples of these residence birds where these resident birds is actually have an occasion where they sometimes mingle around with the migratory birds during landing. So what we did, we also take samples from the resident birds, and we also found West Nile virus RNA from our residence bird. So basically, it is postulated that the transmission of this West Nile virus to our residence birds are basically from the migratory wild birds. And at the same time, what we did also, we take the mosquito samples, particularly the culex mosquito samples uh, from the area which is approximate to the wild birds habitats. So we also found West Nile virus antigen or West Nile virus RNA in these mosquitoes. This is quite interesting. So, but luckily, until now, there is no clinical cases or outbreaks of West Nile virus in Malaysia, particularly in humans. Okay. So other than that, you have also tick-borne encephalitis virus, which is prevalent in Europe, Russia, Asia, as well as in Asia, where the reservoir hosts are rodents, birds, and ruminants. And the arthropod involved is isodis species, and most of the transmissions, apart from vectors, is also from direct injection of raw milk. Okay, so if it's involve ruminant, sorry. So basically, we as I said, we also don't know about the prevalence of tick-borne encephalitis in Malaysia. But however, because most of our ruminants also are imported breed, so there is also a need to study this kind of virus. Okay, we have also looping eel virus, which is prevalent in Europe, characterized by encephalitis in animals, particularly the farm animals. Okay, so this is an example of clinical science in horse. Okay, you will see the horse. This is typical clinical signs of horse infected with West Nile virus. The horse are depressed. They don't want to move. They have hind limb weakness, particularly because it involves the nervous system. Okay. Okay. Next, you have under the genus of pestivirus. Okay. Under genus of pestivirus, you have a virus known as bovine viral diarrhea, classical swine fever, and border disease. Okay, another genus is herpesvirus, but it is actually the human pathogen. So, for the purpose of this lecture, I only cover BVD and classical swine fever. Okay, so you have also border disease virus, which affects sheep, and it is associated with congenital disease, and the prevalence or the geographical distribution is worldwide. Okay, so before I go to the bubonic viral diarrhea and classical swine fever lecture, so basically diagnosis of arbovirus can be done in cell culture. There are quite numbers of cell lines that can support the growth of um, these viruses, particularly the mosquito cell lines. However, most of the time, because these pathogens are classified under risk group 3 or risk group 4 pathogens, just to let you know, just for your information, most of the uh, human pathogen and animal pathogens are being classified into four major groups: risk group 1, risk group 2, risk group 3, and risk group 4. Basically, risk group 1 and risk group 2, you need to perform, you can perform the any works of pertaining to the pathogens in laboratory, uh, in biosafety laboratory level 2, BSL2. Okay, most all of our labs in Faculty of Veterinary Medicine are BSL2. When such as virology lab, bacteriology lab, parasitology lab, because these labs are dealing with pathogens, and most of this most of us are working with research, okay, with pathogens and diagnostic samples as well as for teaching. So most of the samples that we receive from diagnostics are unknown pathogens. So what we do is that we will treat these samples as pathogenic, as pathogens, meaning that because we don't know whether the samples are positive or not. So because of that, we will handle these samples, or in terms of research, handle these pathogens in biosafety laboratory level 2 or laboratory level 2, meaning that in requirement, a main requirement of a BSL2 is that you need to have biosafety cabinet inside the lab. Meaning that when you handle the samples, when you handle the pathogens, you need this biosafety cabinet. You have your lab, and under the lab, inside the lab, you need to have biosafety cabinet. I'm not really sure whether you guys have seen biosafety cabinet or not, but I'm pretty sure later on, when you go your rotation in DVM 4 and 5, you will enter our lab and you will see, you will appreciate like what is the biosafety cabinet looks like. And on top of that, there are certain pathogens that group as group 3 and group 4, which is the effects are more, most of them are zoonotics, no vaccinations available. So because of that, we cannot operate or work with this pathogen in the normal lab. Okay, I say normal lab is BSL level 2. BSL level 1 is actually when you're working with, you are not working with pathogens. Okay, there are some pathogens like E. coli, non-pathogenic E. coli, which you just can culture it on the bench. So that is basically, we can, I can say you can use biosafety level, lecture level 1. But as I said, because most of the time we don't want to have, want to risk ourselves, most of the time the pathogens we will just work it in the biosafety cabinet. So because of that, we work in biosafety laboratory level 2. Okay, in biosafety level 3 and biosafety laboratory level 4, you will need more advanced lab, meaning that the entry towards the labs is actually you have double door entry, and then the air inside the lab is negative pressures, and the PPE that you worn is like the as you can appreciate, like in this now COVID-19 pandemic, the PPE is quite complete. You need to use tire suit, you need to use the respirator. Okay, in the case of biosafety laboratory level 3, and in the case of laboratory 4, the same condition, but in the laboratory 3, your environment is controlled, your PPE is enhanced, and you work in the biosafety cabinet. But in level 4, the environment is also controlled, but you work in the glove box or biosafety cabinet level 3. Okay, you have cabinet and lab. Okay, in level 4 lab, you need to work in the glove box, meaning that you cannot you need to have a cabinet that when you enter your hand, it's like you cannot touch the animals, you cannot touch your glassware and so on. You just operate it in the small cabinets. Okay, I hope that it later on, probably during our lecture, our practical, I will show you what does it mean by level 1, level 2, level 3 for lab, and what does it mean by biosafety cabinet. Okay. And most of the time, these for pathogens are actually based on the classification in the US or UK, Australia, because most of these virus are quite not endemic in their country. So because of that, they operate the virus in laboratory level 3. However, in Malaysia, because dengue, JE, and Westna is quite prevalent and endemic, so most of the researchers just use biosafety laboratory level 2, but with enhanced SOP, with enhanced PPE. Okay, so because in culture, in cell culture, you need to amplify the virus. So because this virus has somehow tropism to the handlers, which is us as a staff or as a postgraduate students, or you later on when you work. So because of that, the precautionary measures or risk assessment need to be take place. Okay, you need to have risk assessment before you carry out this kind of work. Basically, when it involves culturing or amplifying of the virus. Okay. However, as you know, most of the time, culturing of virus is not really enough. You need to somehow have like direct diagnosis such as PCR or RT-PCR. In this case, it's RT-PCR because if you are looking for RNA virus. So having said that, although most of the viruses must be showed or must be visualized or must be confirmed using direct tests, but most of the time for arboviral infections, serological test has been used widely to confirm. And most of the time, the sample that we use is serums or cerebrospinal fluid. This is a good examples of, apart from serums, you also can use CSF samples to detect antibody. Okay, as I pointed earlier in last week or last 2 weeks, you use serum samples for serology, but sometimes you use cerebrospinal fluid. This is true for flavivirus or arthropod-borne virus because the virus has tropism to the brain. So because of that, the antibodies also present in the cerebrospinal fluid. And as you know, serology, you can detect either IgM or IgG antibody, but most of the time for the diagnosis of arbovirus infection, you use IgM. And if sometimes you want to use IgG, you need to use both tests because IgM is produced quite a recent infection. So it's not like IgG, meaning that you just there's no active virus inside the body, so it's just giving you an information that there is the exposure of the virus. And very crucial for this arboviral infection, you need to detect the neutralizing antibodies. Why this is important? Because most of the time when you use IgM specific ELISA, for example, to detect West Nile virus or yellow fever or JE or dengue, most of the time, if the results turn out to be positive, it also could mean, let's say you are using West Nile virus kit. If it is positive, you also need to check for other flavivirus such as the one that I pointed earlier, the yellow fever, the dengue and JE, because these they are envelope proteins. They are serologically or they are related because the host antibodies that reacted with these antigen are the same. So because of that, we don't know. We term this phenomenon as serocomplex cross reaction among this flavivirus family. So how you would like to confirm there's an infection? You need to use neutralizing antibodies technique. So this is just an overview. So normally, if you detect the positive reactions less than before the infections occurs, most of the time it is false positive or contamination. So the moment the infections occur, there will be incubation periods depending on the type of virus. So it will lead to fever, and this is the viremic states. Okay, so during viremic states, because the virus is inside the blood, so you can take blood samples and test for RT-PCR. But frankly speaking, in the real situations, most of us, when we have fever, normally we don't really suspect it is due to viral infections, right? But what I can see the trend now in GH, if you have in general hospital, if you have fever more than 3 days, they will take blood samples. So probably most of the time because they want to check your lymphocyte levels, whether you have viral infections or not. So basically, in terms of arboviral infections, normally RT-PCR is too late if you did RT-PCR, because most of the time most of us or animals have bypassed this viremic period. So as I pointed earlier, IgM-based ELISA are quite common to detect, and if you want to use IgG, you need to couple it with IgM ELISA. Okay, and then you also can use virus isolation in the cell lines and neutralization. Okay, as I mentioned earlier, gold standard for arboviral infection, especially in the serocomplex group of flavivirus, viral neutralization is the gold standard. So basically what happens that you have your serum samples, okay, serum samples from the animals or from human, and you have specific virus that you suspect in the lab. Okay, it can be yellow fever virus, it can be dengue virus, Japanese encephalitis virus, or West Nile virus. So what happens that you mix this virus and the serums, okay, you mix, and what will happen is that when this antibody produce this antibody is specific to this virus, there will be neutralization occur. Okay, neutralization occur. So what? And then you add this mixture of virus and serum into the cells. So this is just an example. You can use Viro cells or mosquito cell line cells, and then ideally, if there's viral infection in the cells, you will see plaque formation. This is because you're using plaque overly agar. So ideally, you will see plaque formation, indicating the virus is replicating. But the positive reactions of plaque reduction neutralization test will be, there will be no plaque formations because the virus already neutralized by the serum. So by using this technique, you can overcome the serocomplex or the cross reactions between the flavivirus. Okay, in terms of prevention and control, it is very crucial to use surveillance, especially in the vectors and the reservoirs animals, to know the status of exposure or to know the status of recent infections. And because this is most of the disease are vector born, so it is highly crucial also to control the vectors. There are a lot of methods you can use, pesticides, cleaning of the breeding grounds. And interestingly, nowadays there is known as Wolbachia project or Wolbachia project. So Wolbachia is actually a bacteria that usually uh, uh, infect the mosquitoes. So what happens that this Wolbachia, it will prevent the mosquitoes to transmit virus. So there's a group of researchers that's working with this Wolbachia bacteria, and they wanted to infect the mosquitoes with this Wolbachia bacteria. And in a layman term, we can say that this Wolbachia will eat the viruses, the flavivirus or any viruses that um, present in the mosquitoes. On top of that, if you read news, if I'm not mistaken, last month I saw there's news in the Europe country, if I'm not mistaken, or in UK. There is a one of the vector control of malaria where they do a they're using what you call it drone. You use drone, and this drone they can sense the I don't know what technology they use, but somehow sense the antigen in the mosquitoes. So what happens that this drone will go to the mosquitoes and kill the mosquitoes. That's very, very interesting, right? Okay, apart from that, vector control, so you can use personal protection such as using long sleeve clothes, screening, bed nets, insect repellents in the house. Okay, vaccination also are available in animals such as in is of course OJE and also in human. Okay, so 11:30. So I would like to can I continue class or you need break?
Can continue, doctor.
Continue, yeah, because you can view the recorded video later on. Okay, so for next we move on into the non-arthropod borne flavivirus. This is under genus Pestivirus. You have bovine viral diarrhea and classical swine fever. Okay, so bovine viral diarrhea is actually an important disease. You will see later on it is a complex disease. I hope that you bear in mind with me. There are basically two genotypes of BVDV, and for each genotypes you have two distinct biotypes, which is known as NCP or non-cytopathic biotypes and cytopathic biotypes. Okay, basically, why this is known as cytopathic or non-cytopathic is based on their characteristic where they can grow in cells and produce cytopathic effects or not. Okay, and most of the 90%, 95% of BVDV of the field isolates the non-cytopathic type. Okay, BVDV is an important cause of morbidity, mortality, and economic loss worldwide in the dairy and beef cattle, buffalo, antelope, carcass, and llamas. Okay, so there are two main clinical manifestation of this disease, as the name implies, bovine viral diarrhea. Okay, it lead to acute uh, typical bovine viral diarrhea. It is characterized by diarrhea. Okay, it is actually acute typical episodic infection of the susceptible animals, and these syndromes of bovine viral diarrhea in certain circumstances it will proceed to the mucosal disease. Earlier, this mucosal disease is actually not classified under BVDV virus, but because most of the BVDV cattle will develop mucosal disease, so because of that, this mucosal disease is also classified under bovine viral diarrhea. Okay, most of the time the mucosal is sporadic, but once it happens, it is really, really fatal to the animals. Okay, these are the worldwide distribution of BVDV. So you can see that the blue legend is actually recently infected after 2008. Okay, and um, the orange is previously infected. You can see in the here some regions of Africa and some regions of America. So and the red legends indicate that there is incidence or prevalence of this disease before 2008 and after 2008, meaning that the area that is pointed in the orange color, they already eradicate the disease. Okay, as for Malaysia, we have a BVDV case, and that's why you can see it is actually recently found. So because of that, it is classified as blue color. Okay, and this is the example. Okay, this is basically the same. It's just that for the white one is the country that there's no data, or the information is quite limited. Okay, so basically transmissions of bovine viral diarrhea involve various means. So it can be vertical transmission, transplacental, horizontal, or vectors borne. Um, as I mentioned earlier, there is this festival is non-vector borne, but there is also some reports, recent reports shown that flies also can transmit BVDV, particularly in the Western. Okay, so for vertical transmission, it is from the contaminated semen where it infects cows, and during embryo transfer to the cow, also by usage of modified live vaccines to the pregnant cows, somehow this virus from the white vaccine, it can vertically transmit and infect the fetus. Okay. For the transplacental, it's actually very clear cut where the pregnant cow is who is persistently infected, it will infect the fetus. And as for horizontal transmission, it involves direct and indirect contact. Direct basically injection and inhalation of the virus. Indirect contact basically the contaminated feeds, equipments, or pets. We term it as fomites. Okay, and there's also one study showed that BVDV can be transmitted between animals during palpations if the same pair of gloves are worn for all exams. So this for all examination of the animals. So this is quite important message to vets, right? So when you handle animals and you suspect uh, there is the infection is you suspect contagious infections, so you need to uh, practice proper biosecurity, proper PPE practice. Okay, and most of the time this virus shed in the most secretions and excretions, and it can survive longer, not really longer, like for weeks in the environment of the animals. Okay, and another means is via vectors, okay, such as flies, horse flies, stable, and face flies. Okay, there are four manifestations of BVDV infection. Uh, Basically, the postnatal infection in the non-pregnant cow, meaning the pregnant cow born, the pregnant cow uh, imagine that you have a pregnant cow, and then it born to a calf that is non-infected. So this postnatal infection occur in the calf that is non-infected from the dam. Okay, they also have in utero infection, persistent infection, and mucosal infection. So what happened in the postnatal infection is that most of the time these calf are born with maternally derived antibody, which is from the colostrum of the mother. So basically, this maternally antibody will protect the calf around like early 6 months of age. So because of that, the pathogenesis or the introduction of the virus was introduced at the age of 6 to 18 months. Okay, this is the typical occurrence of postnatal infections. So basically, the main root is oropharyngeal root, and the primary application occur in the epithelium of the oropharynx, and from there it will be rapidly uptake into drainage lymph nodes, leading to viremia, and it has the tropism to the lymphocytes, leading to leukopenia. Okay, and from here it will spread to other lymphoid tissues such as Peyer's patches. It also have tropism in the alimentary tract, so there will be discrete prominent erosions from the oral cavity into the intestine. So when the lesion occur in the oral cavity, it leads to excessive salivation, and when the lesion occurs to the intestine, it will lead to diarrhea. And for the respiratory tropism, the animals will show evidence of discharge in the nasals and as well as conjunctival discharge. Okay, so basically, the clinical manifestation in the postnatal infections, most of them will recover. The mortality is low, morbidity is quite high. So most of the time, they will manifested as biphasic fever and leukopenia based on the blood profiles. They will have diarrhea, and the nature is like explosive in character, and they have discharge from nasal or ocular, erosions of the mouth, lips, muscles within the oral cavity, and it's also affecting dairy cows. You will see that in the dairy cows there will be decrease in meat yield productions, and you see also profound thrombocytopenia from the blood profile and immunosuppression. Okay. So what happened if the infections started or initiate in the pregnant cows? It's actually will result in several outcomes, which is largely or highly dependent on the transplacental age of the fetus and the strains of the virus that being infected. So basically, the transplacental infection will, if it happened during early pregnancy, most of the time the embryo is infertile, the pregnancy is infertile, or there will be embryonic deaths or embryonic resorptions. Okay, if the infection occur in the mid gestation, there are several outcomes. It can be congenital anomalies of the fetus, such as cerebral hyperplasia. Some of the fetus also may abortion and aborted. Okay, this is typical examples of aborted fetus. And some calf were born, which is apparently normal calves, as you can see here, but they have actually congenital anomalies. If you can see here, you can see that the calf represents white stance, which is typical defect of the nervous system. Okay, normally, if the calf is born uh, born, so most of them will do not have antibody to BVDV. They don't have antibody. So, but they frequently excrete the virus, and therefore they will be the source of infection, and they later on will lead to or develop to mucosal disease outcomes. Okay, and most of the calf born during the pregnancy period, they manifested as weak calf syndrome. Okay, there are actually many causes of weak calf syndrome. It can be nutritional, it can be due to management, and if it is due to infectious, one of it is due to bovine viral diarrhea. It is in the case of BVDV, you can see there will be defects in the eye, lead to retinal dysplasia, in the nervous system, cerebellar hypoplasia, and most of the calf will have like in the integumentary system they have alopecia. Okay, so this is just again to show you what happened during the pregnancy period. If it is happening in the early gestation, it will lead to early embryonic death. If it's happened in the mid gestation, you will have like calf, the fetus will be dead or and aborted, or the calf will born with congenital defects, uh, and they come. There are few outcomes. The calf will be born seropositive, producing antibody to BVD, but not persistently infected. Whereas here you have the calf that could be born persistently infected. Okay, so you have two outcomes. One is seropositive when they have antibody, meaning they are not persistently infected. So meaning this is the type of the type of which one? This one, the non-pregnant or postnatal, because they have maternally derived antibody. So they will be seropositive and they are not persistently infected. Okay. And if infection occur, there will be the new one, the one that is the postnatal infection. However, there is one group of cattle or calf, they will be persistently infected. Okay, what happened in the persistently infected is that they remain infected as a carrier for life. Okay, and most of the time they never mount effective immune response, and they are intolerant to the infections, meaning the immune system of the body will not attack the virus because they already tolerant to the virus. They assume the virus is self antigen. So because of that, these persistently infected calf will be remain seronegative in all standard tests. Having said that, although they are seronegative, they shed large amounts of virus in their body secretion, and they are the source of infection to the other non-infected cattle. Okay, and some of these persistently infected calf will lead to the mucosal disease. Okay, and some of them will, if they are not developing mucosal disease, they are apparently healthy, and somehow, although they are not having mucosal disease, they are the source of infections. So this is the diagram showing what is the role of PI, or persistently infected calf, that constantly shed BVDV. So if these PI calf become infected, they are the one that will cause transplacental infection. Okay, and the cow, this persistently infected can be cow and calf that may have both developed acute infections, and the cow may suffer from infertility. Okay, that can also become infertile. They also can lead to the typical signs of diarrhea, BVDV, such as diarrhea and pneumonia. And it also can be a bull that also will transmit the virus to the female. Okay. So what happens that some of these persistently infected calf will, as I mentioned earlier, will proceed to the mucosal disease. And there is a condition for this infection to occur, for the mucosal types to be manifested, where you need two biotypes of BVDV. Usually it involves the non-cytopathic virus, which is persistently infected in utero, and these viruses are present with the cytopathic virus strain. You call this phenomenon as super infection. If this super infection occurs in the herd, then the calf or the cattle will lead to mucosal disease. Most of the time in the acute phase, there it is fatal. Okay, and in the acute case, you will see severe erosion and ulcer in the mouth cavity, in the stomatitis, and along the intestinal tract. And because of that, they don't want to eat. They become anorexia, and the dehydration and emaciation will be secondary to that condition. And ultimately, most of the mucosal affected cow in the acute case, they will be death, usually fatal. Okay, and in some occasions, some of them will be chronically infected, so the disease will slowly progress with the animals become remaining antibody negative. Okay, this is the typical examples of mucosal disease. So can you guess, class, apart from bovine viral diarrhea causing this kind of lesion, what are the differential diagnosis of cow that can lead to the infection or lesion in the mouth cavity? Can you guess? Class, can you hear me?
Yes, doctor.
Okay. I was talking alone. Okay, so probably because you're tired. Okay, so you need to remember, yeah, if the cow manifested the oral lesions or ulcers, you can suspect FMD. You can suspect infectious bovine rhinotracheitis. I think you have learned it in herpes viridae family, right? There are two types of herpes viridae that can cause the oral cavity lesions, malignant catarrhal fever. You have IBR, infectious bovine rhinotracheitis. Okay, and you have BVD. Okay, so diagnosis of BVD you can make based on presumptive diagnosis, okay, based on the history, examination of the herds, clinical signs, and gross and microscopic postmortem lesion. Okay, so you will see erosion or ulcer along the mouth, through the esophagus, for stomach, abdomen, and intestine. And in the intestine, you will see hyperemia and hemorrhage at the various patches. And histologically, you will see necrosis of the crypt of liver in the enterocytes. Okay, this is the examples of the tongue and the rumen of the ulcerative ulcer that lead to ulcer, and this is the above is the normal Peyer's patches, and this is the affected Peyer's patches with necrotic lesions. Okay, so basically for laboratory diagnosis, you can use if the animals are alive, you can take nasal discharge, feces, blood smear or blood smears, or if the animals are dead, upon postmortem, you can take samples from spleen, kidney, lymph nodes, nasal cabinets, intestine, lung, as well as lung. If there are aborted fetus, you can take the aborted fetus and the fetal liver or kidney. This is basically due to the tropism of the virus. And if you want to take the serological samples, you need to take paired serum samples during the acute and the convalescence period. Okay, you need to take two paired serum samples. Okay, so isolation, you can grow the virus in cell culture. You will see cytopathic effect. So you need to proceed to the either PCR or you can visualize the viral antigens using staining such as immunofluorescence or immunohistochemical. Okay, and for serology, you need to have PET serum samples. So you will see the raise in antibody titer and you will suspect that this is due to BVDV. As for control and preventions, the key of eradication is actually to identify and eliminates the persistently infected infections, which is somehow quite difficult because they remain most of the time seronegative. So other than that, to avoid introductions of persistently infected or the carriers animal to the new herd is by quarantine. What you do in the quarantine is that you quarantine it for a moment of period and then you take blood samples and you observe for the clinical signs. Okay, there is also vaccines available for immunization. They have shown evidence of reducing clinical disease and prevent fatal infections. There are three types: inactivated, attenuated, and live attenuated. However, especially in the term of live attenuated, it can cause clinical infections to the uh, cattle. And most of the time, for the eradication schemes of BBDV regionally or nationally, you need to eradicate the virus without the use of vaccine. This is also true in the case of rabies virus. When you want to declare you are a free state of rabies, you cannot use vaccination. So somehow because but in an area that which is prevalent and endemic, you must use vaccination. Okay. Okay. Last is classical swine fever or known as hot cholera. I will try to finish as fast as possible. So this is highly contagious disease which affects swine worldwide. So before that, I would like to highlight to you guys if you notice the first lectures, the first part of these lectures mainly are involving arthropods, which is zoonotic disease, which is it infect animals and human. And then we proceed to the VVDV and classical swine fever. These are non-zoonotic viruses, but they have very serious effect economically important in the animals population. So, which are classical swine fever and bovine viral diarrhea. So, what happens that this classical swine fever it can affect the domestic pig and wild boar, and it is a severe cause of economic losses, and you need to spend a lot for the immunization and eradication programs. Okay, swine fever is actually prevalent in Africa, Asia. Okay, so basically, as you can see from this map, the green countries are the green that already obtained the free status of CSF, whereas the others are frequently diagnosed with classical swine fever. In Malaysia, actually there are actually sporadic cases, but most of the time we don't really know the prevalence because most of the swine farmers in Malaysia don't want to um, somehow reveal. I don't know. That is based on the what I read in the DVS websites. Okay, and this is the examples of the countries that have eliminated the virus or eradicate the virus, such as US, Canada, Australia, New Zealand, UK, and Ireland. Okay, so basically the transmission of this virus is via direct contact. Also, there is intrauterine, where it also indirectly or formal infections also can occur uh, from the secretions as well as the excretions. Okay, and there's also carriers animals which is inapparently infected, but they will be the source of infections. Interestingly, this disease is also associated with feeding with garbage in kitchen scraps, was which was one time popular. They are the more important mode of transmission. However, this was especially important because many swine were slaughtered when they showed the first sign of disease, and these scraps of the fox, which is containing high titers of virus, will be given to feed the non-infected swine. So this is interestingly how this disease will become more serious. Okay, there's also because of that, garbage feeding and prohibition and garbage cooking regulations are now in place in the many countries to deal with the risk. Okay, this swine fever also can survive in the frozen pork and pork products for years. So that's why you can hear some of the cases like when you travel along the other countries, when you come back to your countries, you bring these frozen products or dried products. This is actually prohibited if you are come from the countries that is endemic for the virus. Okay, in terms of pathogenesis, cholera enter the virus via injection, and the tonsils are the primary sites of infections, and then secondarily it will go to the endothelial cells, lymphoid organs, and bone marrow. And because it has affinity towards the endothelial cells, it lead to profound hemorrhage, thrombocytopenia, and it also affect. It also lead to leukopenia. Remember the bone marrows, the primary organs are the precursors of white blood cells, red blood cells, and thrombocytes. So if these sites are affected, that's why you will see signs of thrombocytopenia, leukopenia, and in this case, because it affects endothelial cells, there will be hemorrhage because the endothelial cells are already being impacted. Okay, it's also lead to atrophy of thymus and germinal centers in the spleen and lymph nodes. This is the common manifestation of acute CSF, where you can see the purpura hemorrhage at the abdomen. So you need to know what is purpura hemorrhage, petechial hemorrhage, and petechial and ecchymotic hemorrhage. Okay, so this is the clinical signs of chronic classical swine fever. There will be most of the time it is atypical clinical signs such as remittent fever, depression, stunted in the growth, wasting disease. It has signs or symptoms of respiratory and gastrointestinal related signs. And virus shedding, this is the period where virus shedding occur, and these herds also are prone to secondary infections, will lead to pneumonia and enteritis. So this is the typical postmortem finding of CSF. You will see here, this is sinuses or bluish discoloration of the ear, a hemorrhage in the kidney, marginal infarction in the spleen, latent ulcers in the large intestine, and you will see this is in the case of bladder, you can see hemorrhage. Okay, diagnosis. Yes.
Doctor, so sorry for interrupting you. Doctor.
Oh yeah, you have that. Okay, so diagnosis, you can read because it's actually the same like the others. Okay, it's just that what I want to highlight here is that you need to know to differentiate between ASF and CSF, African swine fever and classical swine fever. Okay, because they are somehow people tend to confuse. So you just read here what is the differences between African swine fever and classical swine fever. Okay, I think that's all for today. If you have any question, you just can reach me through email or through WhatsApp. And thank you for attention, class. I'm sorry for taking your time. Okay, bye.
Thank you so much, Doctor. Have a
Thank
nice
you,
day.
Doctor. Hey, Doctor.